warahmatullahi wabarakatuh. Very good day to all. Okay, hari ini kita akan membincangkan tentang topik organelle, structure and function that available in our cell. So before we move on to that, I would like to bring our attention to our macromolecules that present in our cell, and each of these macromolecules has its own building blocks. So the first one, we start with sugar. So please bring back our ideas. We have our monosaccharides, and then we have disaccharides and so forth, all right? And then number 2, we have our amino acids. Okay, altogether there are 20 amino acids that you should know. Number 3, we have our fatty acids. And then the last but not least is our nucleic acids. Now, each of these building blocks will build our macromolecules. We have from sugar, what we get is our carb, okay, and the most complex carbs can be present as starch or polysaccharides, or it can be in the forms of cellulose, hemicellulose, and the rest. And from amino acids, okay, combination of these amino acids will get our proteins, and please adhere to examples of proteins that we have. For example, we have in the format of enzymes or hormones, okay. Then fatty acids. From these fatty acids, we can get our lipids, and lipids are the molecules responsible to build up our cell membrane, okay. The last one, our nucleic acid. These are the molecules that are responsible to build our DNA. So now we want to see the relationship between amino acids, fatty acids, as well as nucleic acids, how actually these molecules are being produced in our cell that relates to the organelle presence in our cells, okay. So in order to ensure that we really know the story about organelle and structure as well as their functions, it is very dry if we don't involve ourselves in mapping the idea. So let's map our idea back to the cell structure. So let's draw our cell. Of course, the center of our cell should be our nucleus. So we should draw our nucleus now. Okay. So that is our nucleus, and remember our nucleus is surrounded by nuclear membrane. So I'm going to draw the nuclear membrane here, right? And on the nuclear membrane, we'll find many nuclear pores. So these are the nuclear pores. You should have your imaginary pores in your mind. So I'm labeling, okay, this will be the nuclear pore, and this one will be the nuclear membrane. The whole lot of this will become our nucleus. So what can we find in the nucleus? All right, one prominent element that you can see in our nucleus is what we call as our DNA, that present in the format of chromatin. So what is chromatin, class? It's actually interaction between our DNA strands, okay, together with the complex of protein histones. Alright, so our DNA here. So we want to talk about our DNA, right? This DNA present as double-stranded DNA, and there are two processes, okay, that can happen to our DNA. The first one, the DNA can be duplicated, or in other words, we say that the DNA can be replicated through a process that we call as DNA replication. Next, the DNA also can be served as template in the process to synthesize RNA, which now the specific term synthesize, template for RNA synthesis. So the process of RNA synthesis, the process is known as transcription process. Alright, so how many types of RNA that we have? How many types of them altogether? There are 3 types. The first one is the messenger RNA, simplified as mRNA. The second one is what we call as transfer RNA, simplified as tRNA. And then the third one is known as ribosomal RNA, simplified as rRNA. Alright, so now that is about the nucleus and about our DNA. Next, what else can we find inside our nucleus? We will be able to see another structure, prominent structure known as nucleolus. So this is the nucleolus, right? So what is the function of nucleolus? The function is actually the site for ribosomal RNA transcription. Okay, so the product from the ribosomal RNA transcription will be ribosomal RNA, which later on will combine with ribosomal proteins to produce a structure that we call as ribosomes. And these ribosomes always present in 2 forms. Okay, what are the forms? The one is what we call as large subunit of ribosomes, and we have also small subunits of ribosomes. So whenever the ribosome, okay, next, what is the function of the ribosomes? So the function of the ribosome is actually to perform protein synthesis, or in a specific term, we call that as translation process. Right, next, so whenever these ribosomes are ready to perform translation process, now what will happen? The large subunit and small subunit will combine. So how does it look like? We have our large subunit of ribosome. We have our large subunit and then combine with the small subunit. Our small subunit of ribosomes. Now this will become as what we call as functional ribosomes, means it is ready for translation. Alright, so now we have our ribosomes synthesized in the nucleolus. So next, what will happen? The ribosome will go out from the nucleus, okay, get into the cytoplasm. So I'm going to draw our ribosomes there. So ribosomes, these are our ribosomes, the small and large subunits of ribosomes. Okay, 3 ribosomes. Right, so once we are now out of nucleus, there are 2 prominent organelle that we are able to see. What are they? So the first one is what we call as endoplasmic reticulum. Okay, so that's not the only endoplasmic reticulum class. We still also can find it somewhere here. Right, there are two endoplasmic reticulum. The first one, this one, endoplasmic reticulum, and this is also endoplasmic reticulum. But for the first one, we can find the ribosome studied on the endoplasmic reticulum. So this is what you will see. Okay. So the endoplasmic reticulum with the ribosome now has a new name. We call it as rough endoplasmic reticulum. And the other endoplasmic reticulum, because it does not has any ribosome studied on it, now we call it as smooth endoplasmic reticulum. So what are the functions of this endoplasmic reticulum? Let's start from the left, the rough endoplasmic reticulum. So this rough endoplasmic reticulum, okay, is responsible to synthesis protein. So protein synthesis. Right, and there are two kinds of protein produced by rough endoplasmic reticulum. The first one is what we call as membrane protein, and then number two is what we call as secretion protein. So what does it mean by membrane protein? The protein that is designated to be at the cell membrane. Okay. Protein that are designated to be at cell membrane. And secretion protein means the protein must be exported out from the cell. So example given here is our insulin protein. So that is the duty of the rough endoplasmic reticulum. Now we move on to the smooth endoplasmic reticulum. So now we can see here we give it name as smooth endoplasmic reticulum because the ribosome is not there. So definitely the function of smooth endoplasmic reticulum has nothing to do with protein synthesis. So what is the function of smooth endoplasmic reticulum then? So the first and foremost, the function of smooth endoplasmic reticulum is to synthesize lipids. Or in other words, it is responsible for our synthesis of our cell membrane. Alright, next, what is the function of the smooth endoplasmic reticulum? It is also responsible to metabolize or to break down or to detoxify. These are all the same target, okay? Detoxify. Drugs or toxins, okay? And in our body, okay, there is one particular organ that play this important role. What is the organ class? It should be our liver. Okay, and then number 3, the function of smooth endoplasmic reticulum is to break down or metabolize storage sugar. So storage sugar here, okay, it refers to glycogen. Now, I want to bring back your idea about sugar that present in plants and also in animals. We have our plants and we have our animal cells. Excess of sugar in plants will be stored as starch, and it will be stored at perhaps in the roots or in the seeds. Whereas in animals, okay, excess of sugar will be stored as glycogen. And where does this glycogen will be stored? It will be stored in the liver. So it matches now. Okay, so what happens in our body whenever we are fasting? For example, our blood sugar drops and then we need energy. What happens is next is that the glycogen stored in the liver will be broken down into simple sugar, and later on it will give back the amount of blood sugar required in our blood cells, okay, in our blood. All right. So that is the function of smooth endoplasmic reticulum. Now, what is left now? The last one we should discuss here before we move on to another organelle should be our free ribosomes. So what is the function of this free ribosome? It is, of course, related to protein synthesis. But now, the kind of proteins that free ribosome responsible to synthesis is that should be maintained in the cytoplasm. Should remains in cytosol or cytoplasm. So example of this, okay, we have our enzymes in the glycolysis pathway. So please ensure that you know at least one or two enzymes that involved in the glycolysis pathway so that you can answer examples of protein synthesis done by the free ribosomes, okay? Right, now the business of the rough endoplasmic reticulum has not completed yet, because as we have identified earlier, the protein produced by the rough endoplasmic reticulum is not meant to be remains in the cell. It must be located either at the cell membrane or it should be secreted out from our cell. Okay, example, insulin. So how does this protein move to the cell membrane or secreted out from the cell? So therefore, we need another organelle that can carry the protein synthesized by the rough endoplasmic reticulum. So that organelle is named as vesicle transport. Okay. So protein synthesized from the rough endoplasmic reticulum, okay, will be transferred by the vesicle transport, and this vesicle transport will later move to another organelle. Equally important, the name of the organelle now is what we call as Golgi apparatus. So let's draw our Golgi apparatus class. So that is our Golgi apparatus, this one. So what is the function of Golgi apparatus? It is supposed to, function number one is to receive proteins, okay, from a rough endoplasmic reticulum that's shepherded by the vesicle transport. Okay, next it will modify the proteins. Okay, so what kind of modification takes place here? Perhaps a process that we call as glycosylation. Glycosylation means additional of sugar. Glyco is sugar adding to the proteins. Okay, what else the Golgi apparatus do? It can store the protein. Okay, it is as a storage of the proteins. Okay, and also it can transport out the proteins. So how does the protein after being modified at the Golgi apparatus being transported out? So now comes another organelle responsible from here. So the name of the organelle now is what we call as secretory vesicles. Okay, I'm going to draw that organelle. So this one. Okay. So this organelle later on will move to our cell membrane. So we need to draw our cell membrane, class. I'm going to draw a cell membrane here. Okay. So this is our cell membrane. Remember, our cell membrane is made of lipid bilayer. So let's draw the lipid bilayer here. So the secretory vesicles, okay, carry the proteins that has been modified by the Golgi apparatus, and the kind of modification sometimes involve additional of sugar to the proteins, and these proteins are destined at our cell membrane. So we are going to draw that protein here. Okay, so this is the protein. All right. And through that protein now we can find attachment of sugar. So these are the sugar part. Okay, which now has a new name for this protein. We call that as glycoprotein. The whole lots, okay, we call as glycoproteins. Means our secretory vesicles bring the protein to the cell membrane. So that's how things happen, class. Okay. Alright. Next, what else the Golgi apparatus do? We say that it modify the proteins, it store and also it transport out the proteins. From the Golgi apparatus too, we can find that another organelle being produced. This organelle now, the name is what we call as, okay, lysosome. So I'm going to draw the lysosome for you. What color I should use now? Let me see. Alright, I want to use the light blue color. So this is our lysosome. Okay. So what is the function of our lysosome? Okay, lysosome is responsible, okay, to carry hydrolytic enzymes. So the use of these hydrolytic enzymes is actually to digest food, okay, and also to break down broken organelles. So it's actually for maintenance, okay. So the lysosome remains in the cell, okay, it remains inside the cell. So I guess we stop here for a coffee break, 5 minutes, and then later on we will resume our lecture. All right.